Okay, this is video 3 to go over the PowerPoint presentation for biotechnology with the GFP protein. We're starting at slide number 15. This will surely be our last video. So once you have gone through module 1, which is chemical transformation, where you have the plasmid and the gene of interest inside of the bacteria. That's awesome. You just made the GFP protein, but we're simulating what it's like to make insulin. So how would you get insulin out of the bacteria that's made it? How would you get the GFP from the bacteria now that it's made it? Because we don't want the bacteria, we only want the protein. So in module 2, it's known as cell lysis, and lysis means to break down or break apart. So as it mentions, we're going to be breaking down the bacteria so that we can get those proteins of interest. As it mentions, you would select a single bacteria. You would open the bacteria cell through a lysis solution, which essentially just breaks down the cell through repeated warming and freezing. And it mentions here that in the lab, essentially you'd have like this little pellet that would represent your bacteria, and you would want to make sure that it glows. So if it doesn't glow, that would mean that you were not successful with module 1 chemical transformation. You may have either heat shocked it for too long, or you might not have heat shocked it for long enough. And so that was short module 2. And once you break open the cell, everything's great, but now in module 3 there's protein purification. So for protein purification, we want to get rid of everything else inside the cell. So the cell has tons of proteins inside of it. It has organelles inside of it. We don't want any of that. We only want our protein of interest so that we can, like for example, get the insulin and package it and send it off and know that it's safe and it's only insulin. There's no part of the E. coli involved in it. So we're going to be separating our protein of interest based off its chemical and structural characteristics through a process known as chromatography. And you may have heard this term before. Chromatography is just a way to separate, and separate being a big term there. Separate, in this case, the protein of interest from all the other proteins. So how we're going to do this here is we would have used a column chromatography, separation of mixtures by passing it through a solution or suspension, and so that's an important term you'll want to know. And in column chromatography, as I mentioned, there'll be like these little beads that have certain characteristics to them. The green and blue represents our solution that has the busted open E. coli cells that have all these proteins in it. And so you're able to allow for certain chemicals to pass through while other chemicals do not, and essentially separate the protein of interest in the chromatography phase. So we use a size exclusion chromatography. And so there's these little microscopic pores or channels inside of the beads. And the larger molecules, the more difficult it is to pass through the pores. The smaller molecules can pass through the beads. Yeah. And so this causes a larger, higher molecule weight proteins to move more rapidly. So let me go back to this side real quick. Inside of these little beads, there are little pores. And if it's a larger substance, such as the blue here, they're too large to go through the little bitty pores that are inside the beads. So they move around the beads. The green chemical inside, or green protein inside, they're small enough that they can go through the pores. So as they start to bleed through these beads, they actually also go inside of these pores. And so essentially it makes it a longer trip for these small green chemicals. And so it takes them longer to pass through the chromatography. The blue chemical can just move around the beads, and therefore they can pass through the chromatography more rapidly. So as mentioned, this is known as a size exclusion chromatography. As a quick review, proteins are made up of amino acids, and their structure depends on the sequence of amino acids. And so that can be important on understanding how the protein moves through chromatography and how you can manipulate it to move through chromatography. And it's also important in understanding how we can separate it out from everything else. Just so you know, in the lab, what we would actually do is you would pour your liquid that has your busted up E. coli inside of it and all the proteins inside. And you would see that GFP is a fairly large protein. So it's able to move through this column chromatography fairly quickly. So the blue is representing the GFP, the green fluorescent protein. And what we would do in the lab is we would take a little UV light and we would scan it up and down this column chromatography until we see glowing. And whenever we see glowing, that would tell us, oh, there's our protein. We can actually visually see where our protein is at inside the column, and you would actually see it drip from the bottom here into a beaker, and it would glow. So whenever you would stop seeing the glowing, you would cap this off, and you would not allow anything else to pass through this column chromatography, and all of your protein that glows should be in the beaker. Sorry, I'm going back and forth between slides to help me explain this. These are important. I won't go over them very much, but these are the different structures of a protein. So once we have a beaker full of our protein of interest, the green fluorescent protein, we need kind of a final way to know for sure that's the only protein we have in there. We would not want to think, oh, well, we think we only have insulin. But what if you don't only have insulin? What if you have something else inside this mixture? So we need a way to verify and confirm this is the only protein inside of our mixture. So this is where protein gel electrophoresis comes into play. It's also known as SDS-PAGE. It's very similar to DNA gel electrophoresis with some differences. And so as it mentions, the purpose is to confirm that we only have the GFP protein in our sample, or as we are comparing it to diabetes and insulin production, we only have insulin inside of our sample. We have no other protein. And so I'm going to compare it to gel electrophoresis, the traditional gel electrophoresis of the DNA on the next slide. Not next slide. Next slide after this. So as it's called SDS-PAGE, it's going to explain why. PAGE is that it's a polyacrylamide gel, and it uses varying size pores. So normally in gel electrophoresis, it's one consistent size pore in the agarose gel. These have varying size pores, which makes it difficult for proteins of different sizes to travel through it, and allows for some proteins to travel faster. The SDS comes from, it's a name of a common detergent used to disrupt interactions between amino acids, and it coats the entire protein with a negative charge. That's really important. So as we learned about proteins a little bit up here, they fold based off the way they're charged. And while one amino acid might have a net negative charge, the very next bead or amino acid might have a positive charge. So there's lots of different charges inside of a protein. It's very different than DNA that has a net negative charge. And so how can we get this proteins or different proteins that have totally different charges in totally different areas, how can we get it to move through this gel? And so this SDS detergent makes the protein completely negative, and essentially it returns it back to this primary-like structure. It will no longer have the folds and structures here that are caused by charges, because it gives the entire protein one single net negative charge. So some of the differences. Gel electrophoresis analyzes DNA after it's been cut with restriction enzymes. SDS-PAGE looks at proteins that are not cut, just complete proteins. DNA is naturally negatively charged, and so we don't really have to do anything beforehand other than cut the DNA. With SDS-PAGE, we have to apply that detergent to make the proteins have a net negative charge across the board. Gel electrophoresis is done in a horizontal chamber, which I want to show you. Looks like this. Normal gel lays down, while SDS-PAGE is done in a vertical chamber, which is like this, is where the gel actually stands up and down. Agarose has different size pores, while SDS-PAGE has uniform pore sizes for the proteins to move through. But common things is small fragments in DNA move to the bottom while large fragments stay up top. While in proteins, small proteins move to the bottom while large fragments stay up top, or large proteins stay up top. So in general, on both of them, smaller DNA fragments or smaller protein sizes overall will move quicker down to the bottom of the gel. There is a positive charge at the bottom of the gel for both DNA and SDS-PAGE because both DNA is negatively charged and the protein is negatively charged, so they need that positive charge in order to make it go down to the bottom. So as mentioned, here's how they look different as far as the equipment that you use. You would load it differently too, as a gel would be standing up. Normally you would have your micropipette tip kind of here and you would load into the gel. On this one you actually have to load from the top down. So it's just a different type of technique. It shows here the negative electrode, positive electrode, and how you still have to put buffer into the solution. This is a sample of what the end result of SDS-PAGE would look like. Now, a difference between DNA gel electrophoresis and SDS-PAGE, like in lane 2, if this is DNA, you would say, okay, well they have one fragment of DNA. Versus lane 4, you have lots of cuts to the DNA and lots of fragments. In SDS-PAGE, what this represents in lane 2 is you have one protein. So this, at the end of the lab, is what we would hopefully want to see. We would have one protein, which is insulin. And we would know the size of insulin to know whether or not this is indeed insulin. And so let's say this one is insulin. This is the size of insulin. You can see that in lane 2 it only has insulin. Lane 3 only has insulin. Or in the case of our specific lab, lane 2 has GFP, lane 3 has GFP, and it has no other proteins in it. So that can confirm that our solution is ready to be packaged and sent off for people to use. It has no other protein present. Well, you can see the remainder of these lanes, it has many proteins present. It looks like lane 4 and 5 do not have the protein of interest present because they don't have a band in the right area. While lane 8 looks like it does have the protein present, but it has lots of other proteins in the solution as well, which can be problematic. So that's it. I hope this was helpful. As mentioned, I'm going to put a Google Doc on Google Classroom, so if you have questions, you can write in that Google Doc, and as I have time, I will go through and try to answer those questions in red. See you soon.